Okay, thanks everybody for joining our EECB seminar series. I'm going to go ahead and introduce today's speaker. Dr. Liza Comita is a professor of tropical forest ecology in the Yale School of the Environment. Prior to joining the faculty at Yale, she was an assistant professor in the Department of Evolution, Ecology, and Organismal Biology at The Ohio State University. And she held postdoctoral positions at the University of Minnesota, the Earth Institute at Columbia University, and the National Center for Ecological Analysis and Synthesis. Dr. Comita's work focuses on the ecological mechanisms driving patterns of diversity and species distributions in both intact and human-altered tropical forests. Her research combines extensive field studies of forest dynamics with cutting-edge quantitative analysis to produce novel insights into the processes that drive tropical forest regeneration and also how these diverse communities are structured. Some of her recent studies have investigated how soil pathogens can specialize to infect specific genotypes within populations of trees, thus promoting species coexistence and selecting for increased seed dispersal. Today, Dr. Comita will describe how these and other pathogens can produce diverse forest communities in our seminar. We'd like to thank the sponsoring departments of Forest Ecosystems and Society, Fisheries and Wildlife, Integrative Biology, and Botany and Plant Pathology, as well as the Forest Biodiversity Research Network and the Oregon State University Research Office. Special thanks to Amanda Poley for running the technical aspects of Zoom and YouTube. And with that, I'll turn it over to Professor Comita. Thanks for joining us.
Great, thank you for that introduction. I'm going to share my screen. Hopefully everyone can see that. Thank you, Zach, for that introduction, and thank you all for the invitation to come here. I wish I were there with you in your forests out on the West Coast, but this is a good substitute during these times when we're all stuck wherever we are. So my hope today is to take you on a little bit of a field trip to the tropics. And I know for this seminar series, the focus has been on global pandemics and in the context of the current COVID-19 situation, I'm going to try and provide a slightly different perspective to broaden how we all think about disease and its role. So there we go. So when we think about outbreaks of disease in plant communities, there are these classic examples, things like the chestnut blight that took out a major keystone species from forests in the eastern United States back in the early 1900s. More recently, out in Oregon, I know you all hear a lot about sudden oak death and the way in which this novel pathogen can come in and wipe out iconic species and cause huge shifts in the species composition and potentially diversity of communities. What I'm going to talk to you about today is something a little bit different. It's about how disease is a part of ecological communities, not something that comes in and wipes things out, but rather disease as a mechanism that maintains diversity. So here I'm showing you a picture of a tropical forest. And the first thing that comes to mind when we think about tropical forests is high diversity. So diverse tropical forests can contain hundreds of tree species per hectare. So an area of about 100 by 100 meters can have more tree species than you have in all of the continental US. And how is it possible to have so many species coexisting? And the reason this is a difficult question to answer is the competitive exclusion principle. This idea that if one of the species, one or a handful of the species are competitively dominant, if they are better at using resources, they will push other species to local extinction. So how can you have so many different species coexisting in the community on just a handful of resources that plants use? It turns out there's a whole lot of different hypotheses that have been proposed to explain how you have this high diversity, how you have so many different tree species coexisting. And most of them focus not on what's happening in the beautiful canopy that you see at the left, but what's going on in the dark understory, specifically with these early life stages like seedlings. And much of my work focuses on what's been termed the regeneration pathway, which is you have seeds dispersing away from parent plants, creating a landscape pattern of seed fall in space and time. And then it's interactions between intrinsic traits of seeds, seedlings, and juvenile plants with both the biotic and the abiotic environment that ultimately shape spatial and temporal patterns of plant recruitment, which allow species to coexist. And today I'm going to focus on the biotic environment and how that interacts to shape plant recruitment patterns and ultimately diversity. And in particular, I'm going to focus on a process that you'll hear me talk about a lot today called conspecific negative density dependence, or CNDD for short. And what that simply means is that an individual has a lower chance of surviving when it's surrounded by neighbors of its own species rather than neighbors of other species. So what would cause this? Well, there are two commonly invoked mechanisms. The first is within species competition for resources, that individuals of the same species tend to rely on the same resources and compete more strongly for them. But the second explanation, which has gotten a lot of traction in plant communities, especially in the tropics, is the idea that it's host-specific natural enemies, things like insect herbivores and pathogens that cause this conspecific negative density dependence, and that can maintain tree diversity. So how does this work? There's a classic hypothesis, sometimes called the Jansen-Connell hypothesis, or more broadly, the enemies hypothesis. And it's the idea that seeds disperse away from a parent tree, but most of them tend to land close to that parent tree. So you have high densities of seeds and seedlings close to the parent tree. And those high densities of seeds and seedlings either attract host-specific insect herbivores, or they facilitate this rapid spread of disease through that high density of susceptible individuals. And so I think everybody here now is familiar with the idea of density-dependent disease spread, right? When you have high densities of susceptible individuals and you have a pathogen that comes into the population, it spreads very rapidly. And so what happens is that the chances of a seed or seedling being close to its parent plant are very high, but then it has a high chance of being attacked and dying. And so what that does is it opens up space close to a parent tree for recruitment by other species. Now, seeds that fall far from a parent tree tend to be able to escape from their natural enemies unless that species is very common in the community. Because if a tree species is very common in the community, a seed that disperses far away from its parent just bumps into another tree of its same species and can't escape those pathogens or those insect herbivores. And so seedlings of rare species are less likely to encounter their enemies, and this provides a rare species advantage that allows rare species to persist in the community, and it provides a way to limit common species from taking over. And in this way, this mechanism can maintain diversity. Now, there's been a lot of different studies that have tested this hypothesis, which it's just reaching its 50th anniversary this year. One, in a meta-analysis my lab group did a while back, we looked at experimental studies in which conspecific density or distance was altered. So seeds or seedlings were either put close to or far from parent trees, or at high versus low conspecific densities. And we found overwhelming support, with the exception of the seed stage, where we did not find a significant negative effect of being at high conspecific density or near a conspecific adult tree. And so there's broad support for the idea that being located near conspecific neighbors is detrimental for survival. There's also been accumulating evidence that it's often disease that is causing this pattern. This is just one example. This is a really nice study by Robbie Bagchi and colleagues that came out in Nature a while back now, where they used experiments to demonstrate that pathogen-mediated conspecific negative density dependence promotes seedling species diversity. And so what they did was they sprayed plots in a tropical forest with either water, insecticide, or 2 different fungicides. And what they found in particular with this broad spectrum fungicide that is shown by those red arrows is that when you apply fungicide, it weakens that conspecific density dependent effect. And when you remove those fungi, you actually get a decrease in seedling diversity, showing the role that these pathogens actually play in maintaining diversity of seedlings. Now, it's hard to do those experiments at the whole forest level, but what we can do is use theoretical models, simulations, for example, to look at how these local scale processes driven by natural enemies can maintain high species diversity at the whole community level. I'm showing you just a figure from a paper that you might be familiar with by your own Tall Levy that came out last year in PNAS, and they looked at how tropical forests can maintain hyperdiversity because of enemies. And so there is both theoretical and empirical evidence that natural enemies can cause this conspecific negative density dependence, which in turn helps maintain high species diversity in tropical forests. What I want to talk to you about today is a bit of a variation on that, and specifically looking at how the effects of conspecific neighbors can vary. And I'm going to start off talking about how it varies among species. So how and why do species vary in their sensitivity to conspecific neighbors? Next, I'm going to talk about within species variation, and specifically about a study that Zeb mentioned about evidence for within species pathogen specialization, so specialization at the genotype level. And finally, I'm going to talk a little bit about a study from my lab where a student of mine looked at the effect of forest fragmentation and how that altered conspecific neighbor effects in forests in India. And for all of this, I'm going to talk about it in the context of tropical tree diversity and the role of disease and pathogens in general in potentially maintaining the diversity of tropical forests. So first, I'm going to take you on a trip to Panama. So Panama is in Central America here, and much of my work takes place on an island called Barro Colorado Island that sits in the center of the Panama Canal. This is an artificially created island. When they dammed a river to flood an area to form part of the canal, it became isolated from the mainland. And it's been a research station since then, run by the Smithsonian Institute. And it's classified as tropical moist forest. It has a 4-month dry season from January to May. And it's also home to a 50-hectare forest dynamics plot located right in the center of the island, that rectangle you see there. And within that plot, which is 1 kilometer by half a kilometer, every tree and sapling greater than 1 centimeter in diameter in DBH has been mapped, measured, and identified to species. And they've done this every 5 years since the early 1980s. And so we have data on the distribution and abundance and growth and survival of over 200,000 individual stems. This is just some gratuitous fauna that we're encountering on our trip to the tropics. These are animals that you can see on BCI if you're not too busy looking at seedlings, which is what I spend most of my time doing. So my work has been to work within this 50-hectare plot and look at what's going on with the smaller individuals, specifically with the seedlings and the small saplings. And back in 2001, we established a seedling census in the 50-hectare plot that involves setting up a 1 meter by 1 meter square plot in the center of every 5 by 5 meter section of the 50-hectare plot. So that comes out to a total of 20,000 of these 1 meter by 1 meter square plots. And within these plots, all seedlings greater than 20 centimeters tall and less than 1 centimeter in diameter get a tag. You can probably see it here, those yellow tags, they're actually used on poultry farms, but they hold up really well in tropical weather. So they get a numbered tag, they're measured, and they're identified to species. And we've done this every 1 to 2 years from 2001 up until funding ran out in 2018. And With this data set combined with the data on larger stems in the plot, we have a really good idea of the density of both seedlings and adults of all of the 300 plus species that occur in this site. And using those data, I constructed what I call a neighborhood model of seedling survival. So we can look at an individual seedling, like this one you see here tagged, and we can model the probability of an individual surviving as a function of the density and identity of its neighbors. So we can ask how the density of conspecific neighbors, neighbors of its own species versus neighbors of other species, so heterospecific neighbors, affects survival, as well as how the density of conspecific and heterospecific adult neighbors influence survival. And so we use this data to look at evidence for negative conspecific neighbor effects. Now, many studies had previously shown and continue to show these negative effects, these negative conspecific density effects in both tropical and temperate plant communities. But what studies hadn't really looked at were the variation among tree species in the strength of those conspecific neighbor effects. And so that's what I set out to look at. So do species within the same community vary in their sensitivity to conspecific neighbor density? Or to phrase it another way, does the strength of CNDD vary among tree species within a community? And what I mean by that is, let's say you have species A on the left and species B on the right. And when seedlings of those species are growing without any of their conspecific neighbors around, they each have about an 80% chance of surviving. For species A, if it's growing around with one neighbor, next to one neighbor of its own species, that chance of survival drops to 75%. Species B, on the other hand, when you add a neighbor growing next to it, its chance of survival drops all the way down to 50%. So this means that species B is much more sensitive to negative effects of conspecific neighbors. And so I was interested in how species vary in the strength of those negative neighbor effects. It turns out that they vary widely. So this is a study back from 2010 where we use these neighborhood models to look at this variation. What I'm showing you here is histograms of the number of different species in the community and the strength of that neighbor effect on survival when those neighbors were of the same species, so conspecific neighbors. And for both seedling, when we look at both the effect of seedling neighbors and adult neighbors, we see there's wide variation among species and how sensitive they are to conspecific neighbor density. You'll notice that almost all of the species are negatively affected by when they're growing with conspecific neighbors. But there's a lot of variation in there, especially when you compare it to their response to heterospecific neighbors. So if you ask how they respond to neighbors that are of a different species, you see much less variation, and you actually see the effect is slightly positive. I'm not going to get into that now. If people want to ask about that in the Q&A, I'm happy to ask about it, because it also touches on the idea of herd immunity that a lot of you have heard about in the news lately. But for the sake of time, I'm going to move on. Because what we really wanted to know next was, well, what drives this variation among species and sensitivity to conspecific neighbor density? And the first thing we thought to look at, although actually we looked at it second, but it was the first thing, one of the first things that occurred to us, was species life history strategy. So what we know about tropical tree species is that they vary widely in their shade tolerance. So how well they can survive in the deep shade. And this turns out to be governed by a trade-off between fast growth in high light versus survival in deep shade. And this is a well-documented trade-off that explains a lot of variation in demographic rates within tropical trees. And so we thought it was likely that it also might explain some of this variation among species in their sensitivity to neighbors. So why do we think that? Well, a species location along this growth mortality trade-off axis might influence the degree to which it's impacted by conspecific negative density dependence, because slower growing, more shade tolerant species tend to have higher wood and better defended leaves that leave them less susceptible to pathogen or insect attack. So as part of this life history strategy of having high survival, they invest more in defending themselves against enemies. And sure enough, we found in another paper led by Yan Zhu, who was first a visiting scholar and then a postdoc in my lab, she looked at how conspecific density dependence varies with life history strategy. And we found, particularly at the sapling stage, we found that faster growing species were more sensitive to conspecific neighbor density compared to slower growing species. So the graph I'm showing you here, you can see on the x-axis is conspecific neighbor density, and on the y-axis is this predicted probability of survival. And as you increase the density of conspecific neighbors, survival drops off very quickly for fast-growing species, but much more gradually for slower-growing species. Now, this is just looking at patterns of survival, but there's independent work from other groups that suggest this is related to variation in tolerance of pathogens. So this is a nice study done by Sarah McCarthy-Newman and Rich Kobe working in Costa Rica, where they did experiments to look at species shade tolerance and how well they did in microbial versus control treatments for 21 tropical tree species. So this is the change in performance in a control treatment versus a treatment in which pathogens were present, and found this relationship between shade tolerance and sensitivity to pathogens that we think underlies the relationship we found between conspecific negative density dependence and shade tolerance. So we know that life history strategy explains part of this variation, but another species trait that we thought might be related to this variation is species abundance. And so it could either be that common species suffer more when they're near conspecifics because common species might support more specialized natural enemies that cause density dependence. On the other hand, we thought that rare species might be more sensitive to their conspecific neighbors, which might in fact explain why they're rare. And that's actually what we found. So this is looking at, on the x-axis is species abundance in the 50-hectare plot on Barrow, Colorado Island, and the strength of the conspecific neighbor effect from the other graph that I showed you, this histogram here on the previous slide. And what we found was species that are rare in the community tend to be more negatively affected when they're growing at high densities of conspecific neighbors, whereas species that are more common seem to be almost unimpacted by conspecific neighbors. And this makes sense because if you're a species that does very poorly, that survives very poorly, when you are growing in high density of your own species, then it would be really hard for that species to reproduce, to recruit successfully, and become abundant in the community. Now, again, there's evidence that this relationship is driven by disease, by soil pathogens specifically. So at the same time I was doing this study looking at an observational study looking at patterns of survival and neighbor densities, another group working on BCI was conducting experiments. And we actually didn't realize it at the time. We were asking the same question independently using totally different methods, but we came up with the same answer. So this is work led by Scott Mangan, and he was looking at the strength of plant soil feedbacks, which is another way of saying the impact of species-specific soil pathogens and its relationship with species abundance in the Barrow, Colorado Island forest. And consistent with what I found in my study using observational data for additional species, he found that rare species are much more sensitive to the impacts of species-specific pathogens compared to more common species. And so combined, these two studies suggest that rare species are very sensitive to conspecific density because of host-specific soil pathogens. And this led to an ongoing collaboration with Scott Mangan, as well as with two genomicists, Jim Martin and Claude DePamplis. And Jim Martin had read my study and he had read Scott's study, and he reached out and contacted me and said, I have this idea of what's going on in this system based on my work in genomics in insects. He was previously working with insects. And he had this hypothesis, which is that locally rare species are more sensitive to conspecific neighbor density, because they tend to have a lower diversity of pathogen resistance genes, making them more genetically uniform and more likely to transmit pathogens to nearby conspecifics. We thought, well, that's a really cool hypothesis. And so we went to NSF and we wrote up this big proposal, and we asked for a ton of money to do all this expensive genomic work. And they came back and said, well, you have zero preliminary data, but this is a really cool idea. So we're going to give you some money so that you can start to see if this might actually be supported. And so this was the hypothesis we put together in the proposal, and I'll walk you through it. We think what's going on is actually a feedback loop. So if we start here, unfortunately, I don't think you can see my pointer, but if you start in the box labeled relative abundance, what we know is that small population sizes, species that are at small population sizes, tend to have lower genetic diversity due to things like founder effects, bottlenecks, inbreeding, and genetic drift. And this should lead to lower diversity of resistance genes that control pathogen recognition. Now, this low R gene diversity facilitates the spread of pathogens among conspecifics, because those conspecifics that are susceptible to the same pathogens are the ones that that are more similar in their R genes. And this leads to stronger negative conspecific density dependence for rare species. And this strong pathogen-mediated negative density dependence prevents rare species from increasing in abundance. So they get locked in this feedback loop that they can only break out of if they gain alleles from the regional population to increase our gene diversity. So as I said, NSF gave us some money to test out this hypothesis about a feedback loop, and we got enough money to look at 6 focal species in the BCI plot. I'm showing you the maps of the species here. And we went and we collected seeds and soil from beneath 5 individuals of each of these species. And this is just a schematic of the process. So for each species, we collected seeds from beneath 5 different mother trees. We grew those seeds up in the shade house. And then subjected them to different treatments. We had a sterile soil treatment, and we had a treatment where we applied salicylic acid to turn on our genes, or to induce a response. And we also had a treatment where we applied live soil that would have had the pathogens that attack these species. And then we harvested these species, shipped the material up to Jim Martin at Penn State, where he did a whole bunch of genomics that is outside of my area of expertise. And he came up with this metric of resistance gene diversity. So the allelic diversity of resistance genes for each of these 6 species that we had looked at. And so what we found initially is, if you look here, this is species abundance on the x-axis and resistance gene diversity on the y-axis. What we found, consistent with what we expected, is that locally rare species contain less functional diversity in R genes. So lower allelic diversity of pathogen resistance genes. And it turns out that this lower functional diversity of resistance genes is also correlated with the strength of negative conspecific effects. Excuse me. So we took this metric of resistance gene diversity here on the x-axis, and we looked at whether it was correlated with the strength of conspecific neighbor effects from the previous model I showed you from my work looking at seedling survival, and also with the effect of conspecific soil pathogens from the experiment I told you about, led by Scott Mangan. And in both cases, we found that resistance gene diversity was correlated with the strength of these conspecific neighbor effects mediated by pathogens. And so this provided at least preliminary support for this hypothesized feedback loop. We've since gone back and done a similar study on additional species to try and increase the sample sizes, and that is work that's currently in progress. So I've talked to you so far about what drives variation in sensitivity to conspecific neighbor, or what drives variation in sensitivity to conspecific neighbor density among tropical tree species. And it looks like life history strategy, as well as this relationship between species, local abundance, and resistance gene diversity. But what I was even more interested in is what are the implications of this variation for species coexistence and tropical tree diversity? So this negative density dependence, these enemy effects are often invoked to explain how you have so many species coexisting. But if there's this wide variation among species and their sensitivity, can these effects still maintain tropical tree diversity? And so this is work done, led by a former postdoc in my lab named Simon Stump. I was thrilled to recruit Simon for this project. He's a theoretical ecologist. He did his PhD with Peter Chesson, working on Jansen-Connell enemy type models. What Simon worked on with me is answering this question of what's the implication for the maintenance of diversity when conspecific negative density dependence varies so widely among species. And so he constructed a theoretical model that he drew this nice schematic of for our paper, where you have individual trees on a landscape, they produce seeds, a certain proportion of those seeds are dispersed away, and seeds die due to conspecific neighbor effects. They are more likely to die underneath trees of their own species. And he constructed a model in which species varied in the negative effect of being near a conspecific. And so this is what I'm showing you here is the x-axis is the variation in conspecific negative density dependence among the 15 theoretical species he put into this model. And so the average strength of density dependence here of conspecific negative density dependence is the same for all species. And then he varied how much species differed in that. So when all species have the same strength of density dependence, they're all assigned that same mean strength, all 15 species are able to coexist in the system. But as you start to vary the strength of those negative conspecific effects among species, what we find is that it reduces the number of species that can coexist. So you see these lines going down, fewer and fewer species coexisting. And in some cases, we found that conspecific negative density dependence, when it varies so widely among species, it can actually decrease rather than increase tree diversity. And this was pretty surprising because this is a mechanism that has been invoked frequently to explain how high levels of diversity are maintained. And instead it suggests if you have such wide variation, it can actually lead to local extinction for some species and a decrease in diversity in the community. The different lines in that figure have to do with the proportion of seeds that are dispersed away from a parent plant. And I won't go into that too much, except for the fact that when seeds are dispersed farther away, conspecific density dependence is more effective at maintaining diversity. And so the take-home message from this study is that unless there's some trade-off to compensate for having high sensitivity to conspecific neighbor density, it looks like this conspecific negative density dependence might not necessarily maintain diversity in tree communities. So moving on, just checking how much time I have. Moving on, I want to quickly tell you about work we did looking at evidence for within species pathogen specialization. And this is work led by a former graduate student of mine, Janelle Eck, who's in the photo here in the greenhouse with her seedlings. And we were interested in asking the question of whether soil microbes, soil pathogens, specialize at the within species level in tropical forests. So do we see genotype specialists? Now, genotype specific pathogens are something that are super well known from agriculture, but they've been less well studied, in particular in tropical forests, where we talk a lot about species specific pathogens and how they could maintain diversity, but there hasn't been a lot of work done on genotype specific pathogens. Do they even exist? Do they have an effect? And what are the implications for maintenance of diversity? And so Janelle set up a really nice experiment in the Shade House on Barrow, Colorado Island, where she collected seeds of one species, Virola surinamensis, She actually collected seeds from several other species, but did not have luck with germination. But luckily, this one species, Virola, worked, and she collected seeds from 11 different reproductive female trees. And she grew those seedlings in pots that contained soil collected from either that seedling's mother tree or another reproductive adult of the same species, so another female Virola tree. And in the lab, we referred to this as the mother versus other experiment. And what she found was that seedlings had lower growth in the soil microbial communities of their mothers. And so panel A here is showing the overall effect, the mean across all effects, looking at mother compared to a non-mother Virola female tree. And we found significant reduction in seedling biomass when that seedling was growing in soil of its mother tree compared to a non-parent conspecific. And the panel B is just showing that this trend was pretty consistent across all 11 of these adult trees that we collected seeds and soil from. There's some variation in the strength of this, but we fairly consistently found that seedlings did worse when growing in soil from their mother tree. In addition, we ruled out the possibility that these performance differences were related to soil nutrients or to colonization by mycorrhizal fungi. A master's student in the lab at the time looked at mycorrhizal colonization of roots from seedlings in this experiment, and we found no difference there. And so this suggests that it's highly specialized pathogens that were the mechanism reducing seedling performance in mother soils. Now, this was a really cool result, but it was just for a single species. And we were interested in what were the implications of this at the community level. And so Simon Stump was still a postdoc in my lab at the time. He had just published this other paper where he had this nice model that he came up with to look at conspecific negative density dependence. And I said, Simon, can we plug in genotype specific pathogens into your model and see how that affects tree species diversity? He said, sure, I can do that really quickly. And so he ended up taking the model he had before and modifying it so that it included either species-specific pathogens or pathogens that were specific, that were genotype specific. And so seedlings in this model did worse under their mother tree than under a non-mother tree of their same species. And with that, we could look at the effects of genotype versus species-specific pathogens at the community level. And what we found was that the effects are different for rare versus common species, and I'll walk you through it. So for rare species, genotype-specific pathogens it didn't really matter that much if the pathogens were genotype or species-specific. The benefit of escape from pathogens was similar. So if you are rare and your seed disperses far away from you, it escapes pathogens, whether or not those pathogens are genotype or species specific. However, for common species, and the rare species on this square, the green square you see here, the rare species are in the top row there. For common species, what you see on the bottom row is that they benefit more when pathogens are genotype specific. And that's because a larger proportion of space becomes free from their specialized enemies, especially when genetic diversity is high for that species. And so what you can see in the bottom left there is if the pathogens are species specific, when seeds are dispersed away, they just land next to conspecific adults and they're susceptible to all of those enemies. But in the case of genotype specific pathogens, seeds can disperse away from their parent plant, and sometimes they tend to land near conspecifics, but because they're genetically distinct, they're not susceptible to all of the same pathogens, and they still manage to escape from pathogens despite being common. And so the implications for the maintenance of diversity from this model are that genotype-specific pathogens reduce the rare species advantage, weakening stabilizing effects of specialized natural enemies. And genotype-specific pathogens can promote plant species coexistence in this model, but they do so less strongly than species-specific pathogens. One of the interesting things is we also in this model looked at selection for seed dispersal, so the evolution of seed dispersal, and we found that genotype-specific pathogens select for increased seed dispersal relative to species-specific pathogens. Now, this is really intriguing because, as I mentioned before, when seeds are dispersed farther from parents, when you have more seed dispersal, that tends to make conspecific negative density dependence more effective at maintaining species diversity. So even though genotype-specific pathogens might be less effective at maintaining diversity over ecological timescales, on evolutionary timescales, by selecting for increased seed dispersal, they might then actually increase the maintenance of species diversity further. And so finally, I'm just going to quickly go through, because I know that everyone's been on Zoom a lot already today. I just want to quickly take you to a totally different forest to talk to you about how conspecific neighbor effects might be changing in a changing world. And this is a study done by another PhD student in my lab, Meghna Krishnadas, who graduated a couple years ago now and is faculty in India now, and she worked on plant enemy interactions in a human modified landscape. In particular, she worked in the Western Ghats biodiversity hotspot in southern India in this fragmented forest landscape you see here, which is a matrix of forest and tea plantation. And she was interested in whether edge effects alter the ecological interactions that maintain tree species diversity. So many of you probably already know from conservation biology, ecology classes, that edge effects are what you get when you have small fragments where the forest comes up to a non-forest area that often leads to changes in abiotic conditions, higher temperatures, higher sunlight, lower humidity. And she was interested in how that affects these biotic interactions that are thought to be critical to maintaining diversity. So she set up a series of seed traps and seedling plots along a gradient from edge to interior. So showing the arrow here, she set up, I think it was 15 different replicates where she had plots at the edge and at 3 additional sites moving into forest interior. And what she found was she looked at the diversity of seeds arriving as well as the diversity of seedlings recruiting at these sites. And so on the x-axis there is distance into forest from the edge. So she had sites that were 0 to 5 meters, 20 to 30, 50 to 60, and 90 to 100. And she found no significant difference in the diversity of seeds arriving at these different distances. If anything, it slightly declined as you went from edge to interior. But when she looked at the diversity of seedlings that were recruiting, she found that much higher diversity of seedlings recruiting in the forest interior compared to sites that were closer to the forest edge. And she hypothesized that this was due to a loss of natural enemy effects close to forest edges. And she tested this using a method similar to what I mentioned in the introduction of applying insecticides and fungicides, as well as having a control treatment where she just sprayed water. And she looked at how applying fungicides and insecticides altered diversity at different distances from forest edge to interior. And what she found was 0 to 5 meters, 20 to 30, 50 to 60 from the edge, applying insecticide or fungicide had no effect on seedling diversity. However, at these higher diversity sites in the forest interior, 90 to 100 meters from the forest edge, where we saw the high seedling diversity and recruitment, what she found was that excluding fungi, excluding natural enemies, insects and fungi using insecticide and fungicide reduced diversity. So this indicates that it's actually fungi and insects maintaining diversity, maintaining that high seedling diversity at the forest interior, but those diversifying effects have been lost from the forest edge. Now this is troubling in large part due to the global magnitude of forest fragmentation. This is a study that was out in 2015 by Haddad and others, that found that 70% of remaining forest is within a kilometer from the edge. And even if we look at closer distances, so on the scale of this study I just told you about, 0 to 100 meters from edge, about 20% of remaining forest fragments are within 100 meters of an edge. And so the area where we're losing these potentially diversifying interactions worldwide is huge. So just to sum up, first I told you about variation in conspecific neighbor effects among species, and how tree species vary widely in their sensitivity to conspecific neighbors. And this appears to be related to a relationship between species local abundance and resistance gene diversity, as well as life history strategy. We've also found evidence for within species pathogen specialization. So some evidence of genotype specific pathogens that are ecologically important in these communities. This was, again, a single species study, so we don't know how widespread this phenomenon is, how much it actually matters. But based on that experiment coupled with the theoretical work, the simulations, it suggests that genotype-specific pathogens could be having some really interesting effects in tropical forest communities. And finally, it seems pretty clear that human impacts can alter these plant-enemy interactions that contribute to tropical tree diversity. And so I like to end with this quote, which is, this is a quote from a famous paper called The Empty Forest that came out back in 1992 by Kent Redford. And he was talking about the loss of large vertebrates from forests. And he said, we must not let a forest full of trees fool us into believing that all is well. An empty forest is a doomed forest. And what he meant was the loss of large vertebrates that act as seed dispersers, as mutualists, would be bad for the entire forest, for all of the trees in the long term. I would argue that this concept extends to these interactions with insects and with fungal pathogens, with diseases, that if we lose these interactions between plants and these enemies, that we might have an empty forest that is also a doomed forest. And finally, I was asked, because of the nature and the theme of this seminar series, about what can knowledge from my work contribute to our understanding of global pandemics. And I'll preface this by saying I am very cautious when it comes to extrapolating from my work on plants to anything about humans for various reasons, both biological and just social. But I think there are a few take-home messages about what knowledge of plant pathogen interactions contribute to our understanding of global pandemics during this current global pandemic. One that I think is important is the idea that disease plays an integral role in shaping all ecological communities. It's not just outbreaks when we have a novel pathogen like chestnut blight or like sudden oak death. Disease is a part of all ecological communities and plays an integral role. The second thing, the second takeaway message from my work that hopefully you're convinced of is that density-dependent pathogen transmission results in rapid disease spread when there are high densities of susceptible hosts. In other words, don't go to parties right now. I think that's kind of a take-home message on density-dependent pathogen transmission when we have high densities of susceptible hosts like we do now. And the third thing that I think is really clear from the plant pathogen literature is that people move pathogens around. And now this isn't that closely related to my own work, but about the opening slides I shared with you, which is that the introduction of novel plant pathogens has been frequent and has reshaped ecological communities. And so the idea that we continue to move around novel pathogens with very large consequences as a result, both for ecological communities as well as human communities, is something that I think most scientists have seen coming for a long time. I had this conversation early on in the pandemic with my mother, who said, who would have thought this would have happened? And I said, well, the scientists did, Mom. This isn't surprising based on what we know about disease. And yeah, there's also been a couple Hollywood movies, but there's also been Hollywood movies about alien invasions, which we don't think are going to happen. But scientists were not at all shocked to know that a novel pathogen introduced into a community can have massive effects. And so I'll leave it with that. I'm happy to take questions. I first want to thank all of my collaborators, and in particular, my field team in Panama, who collected the vast majority of this data, as well as former and current students and postdocs that contributed to this work, as well as funding sources. And thank you for your attention.
Thank you so much, Liza. We really appreciate hosting you here. And as we have a few questions more roll in, I'd like to start with a question of my own. So I'm curious about the relative importance of different types of enemies of these tree species. So of course there are fungal pathogens and also insect herbivores, and I'm wondering, does one really play more of a critical role than the other in driving these patterns of diversity and species coexistence?
So is it insects or pathogens?
Yeah.
So it seems to be the classic ecological answer of it depends. Some studies have found a strong effect of pathogens and not so much insects. So the Bagchi et al. Study I mentioned before, they found this strong effect of pathogens in terms of the effect on diversity, but insects had a much larger effect on seedling density as well as the composition of the community. Other studies have found strong diversifying effects of insects or similar for insects and fungi. So it seems to be very context specific. And I would even guess it doesn't just vary from site to site, but it might vary over time as well.
I would like to ask the next question, and this is going to be from somebody who wrote in. So Dr. Comita, first of all, really great talk. And I'm interested, this person wrote, I'm interested in your work on between species variation in CNDD. Have you looked at how the size or distribution changes between forests or forest types? Do forests that have higher diversity have stronger effects, i.e. the Yasuni, Ecuador versus BCI, or temperate versus tropical?
Yeah, so this is an area of active debate right now. There was a paper out a few years ago that used these neighborhood type models. It was a little bit of a different approach, but used these forest plot data sets to look at whether there was a gradient with latitude, whether temperate versus tropical forests differed, whether high and low diversity forests differed. And that paper did find stronger conspecific negative density effects in more diverse tropical forests compared to temperate forests, but there's been a lot of pushback on that study, that it's concerned that what they found were statistical artifacts. It's actually really difficult statistically to make those comparisons because the neighbor densities of conspecifics are so different in these different communities. In that study, they also took a bit of a different approach looking at static data, so not survival, but looking at how many saplings versus how many trees, which is some people have found is problematic. So I think this is still an open question. And there's going to be, some of us are going to be in a working group to try and figure out how you can actually make use of all this amazing forest plot data to answer this question that turns out to be kind of statistically fraught. Yeah, so I'm hoping we'll have a better answer to that when we can have a workshop, when we can actually all get together. But I am agnostic at this point.
Next question here from Abigail Neat. Do you expect to find a higher potential for forest blights and other infections on the edges of forests where the conspecific negative density dependence plays less of a role in maintaining species diversity versus the interior?
That's a really interesting question. You know, what we found suggests that, you know, the disease impacts, at least on seedlings in the understory, are lower near the forest edge, at least for specialized pathogens. There is some evidence that generalist pathogens and generalist insect herbivores might be more common in fragmented forests and could have a larger effect. And so the relative effect of generalist versus specialist pathogens really makes a difference if you're thinking about the maintenance of diversity. But from what we know about kind of outbreaks from tropical forests is pretty slim in terms of the data available at this point. But that's a really interesting question. Not a great answer, but an interesting question.
I will ask the next question here, which is about R genes, and it asks, Liza, is the diversity of R genes equal to heterozygosity or to number of R gene loci? In either case, is the R gene diversity correlated with diversity in other host genes?
This is what I was afraid of. I told Andy, I was like, I always get nervous when people ask me detailed questions about the genomics because I always defer to my genomics collaborators. What I can tell you, and I might be changing the meaning of the question a little bit, what we found were these relationships between abundance and strength of conspecific neighbors with R gene diversity. We did not find them for that same relationship with genetic diversity in the remainder of the genome. So it looked to be specifically a relationship, not just between overall genetic diversity of the population, but something specific about resistance gene diversity, if that answers the question.
Yeah, thanks. Here's another question, actually from the paper that we read for our seminar series course. In the mother versus other study that your lab conducted, do you know how closely related the seedlings were to the other, the non-mother conspecific trees that they were grown other?
Yeah, so we ended up collecting, so initially that wasn't part of the study just due to lack of resources. Towards the very end of our PhD, Janelle managed to get a dissertation improvement grant from NSF, and she had smartly stored some of, when she harvested the experiment, stored some of that tissue in freezers in Panama. And then she ended up going to Jim Martin's lab and learning these genetic techniques and genotyping those seedlings. We don't have those results yet. It's been a long process, but we will have that information, which will be really interesting because we'll know how related those seedlings are to the mother tree, but also how related they are to each other. And this experiment was made possible because this is a dioecious species. So we knew that the mother tree had to at least supply half the genetic material, and that other mothers couldn't also be parents. And so that simplified it a bit, but it would be really nice to know exactly how closely related each seedling was to the soil source. And so we do have those data and they're being analyzed, but I don't know the answer to that just yet.
The next question here, hopefully we're not too over and we can still talk. The next one here asks, has the prediction that pathogen populations are host genotype specific in these plots, not sure which plots, in these plots been tested? Evolutionary pressure on the pathogen populations would select for host genotype non-specific pathogens.
Sorry, I keep muting myself to avoid feedback. So evolutionary pressure on the pathogen populations would select for host genotype non-specific pathogens. So here's the thing about the way I think about it, that originally I didn't, and then I was talking to a plant pathogen, a well-known plant pathogen ecologist, who pointed out the difference between lifespan in trees versus pathogen communities. And we were talking about it in the context of succession, that you have a field or a deforested area, and then you have a tree that gets a seed dispersed in there, and that seed grows up. And I said, well, it would take a long time for that species specialized pathogen to find it, so it would escape. And he said, think about it. These are tree species that live, decades to centuries. And we're talking about soil pathogens with much faster generation times. Over time, you're going to get things that specialize, that are already there. And so I think that it isn't unlikely to expect very specialized pathogens to be occurring beneath a large tree, because there's been enough time for those populations to evolve and to prosper at that site occupied by that individual tree. And it's too bad that this is like just a Q&A, because a lot of these are questions that would be more conducive to having a back and forth conversation. I don't know if you buy that, but I do think that it is entirely feasible and possible to have these highly specialized pathogens that occur beneath a single host tree, just because of the difference in lifespan.
Thanks, Liza. We'll finish with one more question here, since it's 5 after. So Tal, whose work you cited in your talk, posed a question. We have also found that variation in sensitivity to conspecific negative density dependence, so CNDD, erodes diversity, because species with the weakest density dependence have a huge advantage and other species are pushed to extinction. So if that's true, then how do rare species persist in the community without that rare species advantage? Do you think the strength of CNDD is dynamic, so that the pathogens are increasingly evolving to target common species?
That's a great question. And when I first presented my work on differences among species in strength of density dependence and rare species being more sensitive to conspecific neighbor effects, I kept getting this question of, well, how are they even in the system then? So one answer that I hinted at towards the end was it could be that it trades off, that species that suffer strong CND, that are very sensitive to conspecific neighbors, might actually benefit, might have some other characteristic that trades off with that and balances out that negative effect. And so one thing, actually, we had a follow-up paper that unfortunately got buried in a purely theoretical journal, and so I think a lot of people didn't see, was this idea of testing whether there could be a trade-off between dispersal, how far or how many of the seeds of a species are dispersed away from the tree, versus how sensitive it is to conspecific neighbors. And this makes perfect sense to me, because as we found before, that being very sensitive to conspecific neighbors selects for more seed dispersal. And if you have a species is able to disperse its seeds further away, then more of those seeds escape. So if it's more sensitive to conspecific neighbors, but it's better able to escape those neighbors, then that would balance it out. And so I think that is one potential trade-off that might explain why these more sensitive, these species that are more sensitive to conspecific adults just haven't completely been pushed out of the system yet. The other answer in these communities is always, well, maybe they just haven't been pushed out of the system yet. Yeah, but it's definitely an interesting question that I would love to know the answer to.
Thanks, Liza. And thanks everybody for tuning in. And next week, we will have another seminar talk at the same time.
Great, thank you so much.
Thanks so much, great talk.
Thanks, Liza. Can you stick around for a couple minutes? There's a couple questions that the organizers had, including me, that we
Oh man, you're all gonna grill me now?
No, just stick around and don't go.
Oh man. Keep her forever. We exist in Zoom.
It's true.
I'm looking at all of these questions now. Oh, that one question came from Alan Harry.
There were quite a lot of people who heard about this. I saw Carlos Perez was on here.
Well, Andy blasted it out to the Forest Geo.
Send it out to the Forest Geo serve, yeah.
Is that why there's so many people on it?
Today, which that makes it a bit of a different audience. I'm trying to figure out who asked that latitudinal variation question, if I made anyone real mad.
Dale Forth.
Thanks for your talk. That was really excellent. I have to sneak away, but I also really appreciated the way that you answered the questions. I wish that there had been more back and forth. I know that we're like restricted.
I'm like, do you? I don't know if you believe what I'm saying because I'm making most of it up. It's like,
no, you didn't seem like you were making it up at all.
Oh, but good. I've learned to fake it.
I wonder if we could unmute some speakers, have a little back and forth, like when we know they're not spammers or something, and they've asked an interesting question.
Yeah.
I have to also go to another meeting, but so I really like the idea of if your exclusion zone is big, you can